Good morning. So today we're going to talk about the immune response. And part of the reason for this that we're doing this is to get you ready for the immune response exploration that you'll be completing over the next couple of days. Really, the only needs you have today are internet access and to be aware of our learning objective, which is explaining how our bodies respond to invading pathogens using evidence from previous diseases and how it relates to the current COVID-19 pandemic. So, um, your task for today, basically, all you really need to do is watch and follow along with this lecture. And then, as far as submitting things, you don't have any homework to submit tonight. However, the immune response exploration will be posted, and you could technically get started on that if you wanted to. It'll be probably a multiple day thing. So here we go. So yesterday, or excuse me, yeah, I guess it was yesterday. Yesterday we talked about the different types of pathogens. We had bacteria and viruses and fungi and parasites and prions. And so all of these things, all of these pathogens could potentially infect you. They could potentially make you sick, cause you injury. And so the question is, how in the world do we defend against all of these things that are trying to get at you or in you? And your body's pretty amazing in that there are two huge categories of defenses that you have that protect you against pathogens. And they are either ones that you already have. They're called innate defenses. Okay, an innate skill is one that you have, or innate knowledge is something that's kind of internalized. It's something that you don't have to work at. So innate defenses are something that's just there. All right, and then there's acquired defenses, which we'll talk about specifically shortly. So as far as innate defenses go, these are not specific to any particular pathogen. They are more general, and they will counteract the effects of many pathogens. All right, and these things include barriers to entry. So if you have intact skin, or if you have, if the barriers that are there naturally in your body are intact, then that's one of your innate defenses. And you also have phagocytic white blood cells. Phagocytic means cell eating. Okay, phago means to eat, cyte means cell, ic means pertaining to. So these are white blood cells whose job it is to eat things. All right, and then basically they scan the area looking to eat anything that doesn't belong. All right, so as we talk about barriers to entry, things that would be included in that list would be intact skin. Okay, it has to be intact skin. This is one of the reasons that if you get a cut or a scrape or a blister or any of those things, that is a breach to your skin. And so that would not be intact skin, which then means you're open for pathogens to enter, whether it's a bacteria, virus, whatever. Mucous membranes, those are the moist membranes inside your nose and inside your mouth, and various other orifices. All of those mucous membranes contain white blood cells that are there looking for invading types of things. The mucus also traps it. There are enzymes in there that help break down any kind of pathogens, and it's pretty efficient that way. Chemical barriers. Okay, in your sweat, saliva, and tears, there's this enzyme called lysozyme, which lyso means to break, destroy, or rupture. And it's an enzyme that breaks, destroys, and ruptures the bacterial envelope, the protective part of the bacteria. So it's actually an antimicrobial type of an enzyme that's in your sweat, saliva, and tears naturally. And then if you eat things, you have stomach acid. Okay, your stomach pH is like 2, which is a very, very strongly acidic environment. There are other strong acidic environments in your body, but that's the strongest area. And so if a bacterium or virus gets into your stomach, unless it is extremely hardy, it is probably going to be broken down by the strong pH in your stomach. Okay, other resident or innate defenses, you have bacteria that live inside of you in your mouth, your nose, your intestines, and the vagina, and the rectum, and various other places that are basically they use what's around them, and their waste products kill off other pathogens. Okay, which is why when sometimes people are on medications that disrupt their natural flora, that's one of the words used. Flora is like the bacteria that live in you and on you. If that's disrupted, it can cause other opportunistic infections to take place. Okay, we talked about yeast infections yesterday. There are other ones just like that. All right. As far as, so that's the end of innate defenses. As far as acquired defenses, if you acquire something, that means you get it from somewhere else. If you acquire some money, you either earned it or you stole it, right? Or if you acquire a car, hopefully you bought it and you didn't steal it, or somebody gave it to you. Okay, so acquired defenses are ones that you receive somehow. All right, you're exposed to lots and lots of these things we call antigens. Antigens generally are things that don't belong in you. It's little chemical markers on the surface of pathogens that your body recognizes as foreign. Okay, so your normal human lifespan, you're going to encounter millions of antigens, which means you're going to have millions of these cells, these B and T lymphocyte cells. Okay, we haven't really talked about these yet. That's part of the point of your immune response exploration that you're going to do is you're going to learn all about B and T lymphocytes. A lymphocyte, lympho refers to the lymphatic system. All right, cyte means cell, so it's lymphatic system cell. We're going to talk more about the lymphatic system next week, but just know that these are cells that are associated with the lymphatic system, and they are there to help defend you. Okay, they produce antibodies. All right, and each antibody will respond to exactly one type of antigen because antigens are chemicals, they're oftentimes proteins, and they have a very particular shape. And so an antibody has to have that exact same complementary shape in order for it to bind. When they do bind, then it signals other white blood cells to go to work to destroy whatever it is that they've bound to. There are various ways to acquire defenses, okay, and we can basically drop these down into two categories. We have active immunity, all right, where you have, let's say, had the chicken pox. I'm guessing that many of you have never had the chicken pox because they started doing the vaccine for chicken pox a number of years ago. And so most of you had the vaccine. All right. But I had chicken pox when I was in third grade. I remember it well. It was very itchy and awful. But my body has been exposed to that virus, the chicken pox virus. And so I have antibodies in my bloodstream that if I ever encounter the virus again, they should be able to fight off and I'll never get chicken pox again. Okay. So that'd be active immunity. You can also gain active immunity from vaccines. We'll talk about those specifically here in a minute. Passive immunity is where you have antibodies, the antibodies that your body could make, all right, that have been extracted from somewhere else, are injected into you or added to you to elicit a response to fight the infection. So it's like artificial type of immunity. You're just getting the antibodies that you didn't make, but something else did. Okay, we're smart. The human species is pretty smart. We figured out ways to actively initiate the production of antibodies and T and B memory cells through vaccines. Okay, vaccines. Basically, the original vaccine was made by Edward Jenner, I believe his name was, and it was against smallpox. And what Edward Jenner discovered was that milkmaids who had a disease called cowpox, which was common among milkmaids, if they had that, they couldn't possibly get smallpox. And so what he did was he took some pus from a cowpox lesion and he injected it into someone else. That person got cowpox, and then he exposed that person to smallpox and they did not get smallpox. Now, this was like close to 150, 200 years ago. You could never get away with this today. You'd be sued. But that's how vaccines really got started, was with a smallpox vaccine made by Edward Jenner. Basically, that was an aside. Sorry about that. What you can do, what they do now generally, is they will take a dead or weakened form of a virus that has the same antigen, the same thing that your body's going to respond to as a real virus. They'll inject that into you, and then your body responds as if it's actually fighting off the real infection, producing antibodies that can then destroy whatever that antigen is. And once you have those antibodies, you keep them. Okay. Now there are some diseases like tetanus and various others where you have to get boosters every so often, because if you get it, your body takes a while to respond. Those antibodies are there, but it basically just boosts your body's defenses. Okay, but vaccines are there. That's what they do, is they initiate your resistance to a disease artificially, so to speak. Okay, there's been controversy. Okay, and I'm not going to get in. We're not looking to debate this with you because it's been well and thoroughly debunked. Okay, there were some papers published. It's been at least 10 years ago that suggested there was a link between vaccines and autism. Okay, those papers, every single one of them has been retracted. Okay, they've been taken out of journals. They have been removed from the research, from valid research. Because a lot of the data was falsified, the links that they found were tenuous at best. However, that research was published, the damage was done. Shots are scary. Okay. And so people sometimes have chosen not to get vaccines just because they think that, they're going to give their kids autism. It doesn't happen. It doesn't happen. So the problem with this is that there are pockets of the United States where diseases that had been eradicated in the United States are now back, like polio and smallpox. Up in the upper Northwest, there was a measles outbreak up there. Measles should not be a thing in the United States. It's been basically eradicated, but it occurs in these pockets of people who are anti-vaxxers who end up getting these diseases, and they pass them to one another. It's just a bad thing. So get your vaccines. They're not going to kill you. Okay. Other acquired defenses that you can get. So sometimes they will identify an individual who has had a disease, has been exposed, who then has antibodies, and they can actually take a sample of their blood and they can isolate those antibodies and then inject them to other people. Okay, that's one option. One of the ways they can grow antibodies is they can grow them in vitro, which technically means in a test tube. They can also be grown in humongous vats. All right, but basically they will find usually bacteria or yeast that can produce antibodies based to a particular antigen. They will grow them. They'll use mouse lymphocytes, and then they can extract these antibodies and use them to treat a disease. And then finally, they can grow them in vivo. One of the things they do, they grow them in chickens. And what they'll do is they'll expose chickens, and then they'll harvest their eggs, and they'll get antibodies out of their eggs. They can also grow them antibodies in cows, or not cows. Sorry, excuse me. The most likely ones are goats and sheep, where they'll inject them with a vaccine or a weakened form of the disease, and the goat or the sheep forms antibodies, and they'll take samples of their blood, and they'll be able to take the antibodies there and then inject them into other people, and that can help with acquired immunity as well. Okay, with all of these immune things, sometimes there are issues, and some of the big ones that we're going to talk about. First of all, if you have allergies, I have allergies. Okay, I'm allergic to shellfish. I'm allergic to other fish. Anything that swims, I really can't eat. I discovered this when I think I was 6 years old. My dad was in charge of dinner one night because my mom had something going on. And so he took the easy route and threw some fish sticks in the oven. Okay. And I started eating the fish sticks, and I didn't love fish sticks, but I was eating them because my dad was in charge. And so I was eating the fish sticks and my tongue started to itch. And I was like, I was like, I don't like this. And I told my dad, I said, my tongue is itchy. And he said, just eat the fish sticks, like dads do sometimes. And so I ate the fish sticks. Well, after dinner, I went into the bathroom and put the seat of the toilet down. And I sat down because I didn't feel good. And I was sitting there. My older brother came in, who I talked about yesterday, who got salmonella. My older brother came in. He took one look at me. He's like, "Dad," and he called my dad in. Well, my lips had swollen up, and I had hives all over my face, and I was breathing with like this super high-pitched wheezing. Okay, that's an allergy. And so what happened was my dad basically poured a bottle of Benadryl down my throat, and I recovered. Now, I did have another reaction later, and after that, then they suggested that I should probably keep an EpiPen around in case I had a reaction, because it just gets worse over time. Anyway, that's an allergy. Okay, so basically what happens is there are antigens on the surface of harmless items. In my case, things like fish, there are proteins in there that my body, just for whatever reason, overreacts to. Okay, so what happens is that you release these things called histamines, and histamines initiate what we call the inflammatory response. It causes itchiness, it causes swelling, it causes increased blood flow to the areas that are offended, which could be places like your lungs and your throat and your mouth and face and all of those kinds of things. All right, Benadryl and various others like it are antihistamines. They try to counteract the effect of histamines. If you have seasonal allergies, you're probably taking an antihistamine. All right, so it helps block the histamine response to those allergens that really shouldn't really be affecting your body like they are. It's just an overreaction. So immediate allergic responses, you have antibodies that go to work right away, and they react to the allergens that are in your body. Anaphylactic shock. Okay, this is life-threatening. People who are allergic to things like peanut butter and bee stings and any of those things who have anaphylaxis, that causes basically, it's called a guarding. Anaphylactic means a guarding against, and it's a guarding against this allergen. Well, the problem is the guarding is an overreaction, and it closes off your airway, and it can kill you. All right, which is why people who have severe allergies to food and bee stings and things should have an EpiPen with them. And then there's delayed allergic responses. These are initiated by memory cells, memory T cells, memory B cells. These are ones that are part of your body, and then they can ramp up their response over time. Other immune problems, organ transplantation. Okay, we have your organs are full. So if let's say if you're getting a transplant of a kidney, okay, kidney transplants are pretty common. Mr. Sulo has a third kidney because he had a kidney disease that rendered his kidneys almost inoperable. So I think his sister donated him a kidney. Anyway, organ transplantation, your organs are made up of lots of things. They're made up of oftentimes connective tissue and epithelial tissue and various like muscle tissue perhaps. And so all of those different tissues and all of those cells have lots of antigens, little proteins that stick out of the surface of them that identify them. They're like little name tags, like I'm a kidney cell, I'm a heart cell. Okay, and so there's lots and lots of them. The problem is that if you put an organ that's not yours into your body, all right, the antigens on that organ are going to be different than yours. Okay, so what has to happen, first of all, during organ transplantation is that they have to very carefully select the donor. Okay, if they select a donor that's very similar, which is why you oftentimes select a sibling, all right, that'll reduce the amount of immune response to that new kidney. You can use immunosuppressive drugs. All right, I know that most people who are organ transplant recipients have to stay on anti-rejection drugs and or immunosuppressive drugs their entire lives. Otherwise, your body will attack that organ and cause damage, and then you're back where you started. New and emerging types of things. Genetic engineers are trying to make organs from similar organisms. Pigs, we've transplanted things from pigs like heart valves and things like that for years. What they're trying to do is engineer organs in pigs that equivalent to the type O blood. We haven't talked about blood yet, but type O blood we refer to as the universal donor. If you're dying because you're bleeding to death, if they give you type O blood, you're not going to have a reaction to that because it doesn't have any antigens. So the hope is that they can do that with organs, then people won't have as big a reaction to new organs being put in. That's called xenotransplantation. And then finally, they're trying to grow artificial organs. One of the bits of research I read is that they're making frameworks with 3D printers and then trying to stimulate the growth of cells on those frameworks into new organs. And those new organs would be without many antigens. All right, so then they won't have an immune response. Whew, I've been talking a lot. So here are a few review questions. Once again, I'm going to go read through them. You can scroll back through the slides that we've just covered and kind of get an idea of what these mean. You'll get more information on these as we go forward with the immune response exploration. But basically, here's the questions. How are T and B lymphocytes similar? And how are they different? Okay, so here's what you're not going to say. T and B lymphocytes are similar because they're both lymphocytes. Much too easy. Okay, there are some structural and other types of similarities. So look a little deeper when we get into this exploration. What are antigens? What technologies do we have to boost the human immune system? What are vaccines and why is there controversy surrounding vaccines? Okay, there's a few other questions that we'll get to at another time, specifically related to the lymphatic system, which we'll cover next week, probably on Tuesday. All right, so specifically, as we move forward, now this is the lecture part is done. There are a few more slides that we're going to cover that specifically deal with the immune response exploration that you're going to be starting. Okay, so for the immune response exploration on this slide, what you'll see is on the left-hand side of the screen, you have innate immune system components. On the right-hand side of the screen, you have adaptive immune system components. And in the middle, those kind of overlap, okay? So on the left-hand side, dendritic cells, neutrophils, macrophages, basophils, eosinophils, and natural killer cells. Okay, those are all cells that are already present in you. They are not specific to one type of antigen, and they are circulating through your body in various locations looking for foreign invaders and trying to kill them. Okay, on the right-hand side, you got B cells and T cells. You can see that on those B cells and T cells, that I'm going to just put the cursor around it. There are these surface antigens here. Those are like antibodies, and they have particular shapes. All right, this is a very simplified version of what they would really look like, but they are particular to an antigen, and they will only respond to one particular type of antigen, which is why you have so many different kinds. All right, so B versus T cells. B cell receptors bind to any antigens free or on the surface of a pathogen. So you can see that over here to the left. Here's an antigen. All right, this antigen, it could be, I don't know, it could be a bacterium, it could be a virus. As long as you have a B cell with a receptor that fits that antigen, all right, then it will bind. Okay. If you have one that does not fit, like for example, this antigen is kind of square shaped, it would not fit in this B cell receptor. And so this B cell would ignore that one. All right. T cell receptors bind to antigens present on the surface of host cells. So these are cells that have probably been invaded by some antigen, or a macrophage of some sort has engulfed an antigen. We'll talk about that in a minute. And so the T cells come along and they're looking at the surface of cells and they see, oh, here's an epitope, which is part of an antigen. It's like the protein part that sticks out, and it recognizes it and it attaches. Okay. So B cells and T cells, the way they bind is a little bit different. That would help you with the question on 2 slides ago. All right. Other things, okay, innate immunity and how it morphs into adaptive immunity. You can see in the left-hand side of the picture, you have some sort of a microbe. We'll just call it a bacterium. Okay, this bacterium gets into you. You have a macrophage, which is a white blood cell, which recognizes this as not being part of you, and so it engulfs the bacterium. Okay, having done that, it will then turn into what we call a dendritic cell, where it will take an antigen from the surface of the bacterium and put it to the surface of itself. All right, a helper T lymphocyte comes along and says, oh, that doesn't look good. All right, which activates helper T cells, and then it can kind of branch off from there. You have B cells that come into the equation, and then antibodies get produced, and then you start killing off this bacterium. You can see down in the lower right-hand corner here, these antibodies here have been produced by the B cells, which become plasma clone cells, and you end up with all of these antibodies which attach to the bacteria, which kind of inactivate them or keep them from doing the bad stuff that they could be doing to you. And this will become more clear as you go through the exploration. It's a much more in-depth. All right, so antigens versus antibodies. All right, on a bacterial cell, this is a really nice illustration. Here on the right-hand side, you have this bacterial cell, and sticking out of the bacterial cell, you have various proteins. They're called epitopes. Epi means on top of. All right, epi means on top of or on the surface. All right, and so these epitopes, they sit on the surface of the bacterium, and they're kind of like identifying their name tags for the bacteria would help identify what it is. All right, if you have antibodies that fit on that, it will attach. Okay, you can see how this one that's shaped like a Y here attaches to this epitope. All right, they attach to the antigen. The antigen is the thing that's irritating. It's the thing that's not part of you. It's the identifying feature of a cell. The antigen is the exterior identifying feature. And so the antibodies specifically attach to particular antigens. All right, they're specific. They're not just general. And then finally, I think this is the last slide, the cell-mediated immune response. So here's a macrophage. This is from a couple slides ago where it's ingested. It's actually encountered a cell that was infected. Let's say it was infected with coronavirus, okay? So the coronavirus has invaded this cell right here. This would be like a lung cell, all right? And the coronavirus is in there, and the cell is showing characteristics, because one of the things the coronavirus does is some of its antigens break off and move to the surface of the infected cell. All right. And so this macrophage identifies that this cell is not normal, and so it engulfs it. Okay, what the macrophage then does is it takes some of those antigen complexes and pulls them off of the infected cell and puts it in its own cell membrane. And then you have the T cells come in, and there's a lot of mitosis that goes on. And it basically, the course of it is they attach the macrophage, they reproduce, and then they start to kill the cell that contains the virus. Okay, this is the ideal situation, and your body will do this to coronavirus cells, which is actually, if you watched the video a couple of days ago, one of the reasons that your lungs get pneumonia is because you have this buildup of dead cell parts and things like that, which can lead to pneumonia, which can lead to the bad complications that are associated with corona. Anyway, one of the chemicals that's in charge of all this are cytokines. All right, and this is a class of chemicals. They're secreted by certain cells in the immune system, and they have an effect on other cells. And so one of the things they can do is they can stimulate memory T cells. They can stimulate the attraction for killer T cells. There's all kinds of things that cytokines can do. Okay, let's go. That'll work. So there's a lot to it. This is a very, very basic overview. If you were an immunologist, one who studies the immune system, you would look at this and be like, oh yeah, that's very simple. Okay, it's a very simple explanation for what's really happening, and that's okay because we're not going to be immunologists after this unit. We want you to have a good understanding of how immunology looks, and also how your lymphatic system is involved with this. Okay, so we'll see that as we go forward. I hope that you all are getting something out of this, and we're going to start connecting it specifically more to coronavirus in the coming days. But this is all kind of the background information before we can understand all of that. So, hope you're having a great week. Get out and enjoy that weather while it lasts, and we can't wait to see you guys again. We still hope we get to see you this year.